Biochemistry 401 Lecture 38. Today we're going to talk about transcriptional regulation, and we're going to compare and contrast regulation in E. coli and in eukaryotes. We're going to talk about the cis and transacting elements and classes of transcription factors. And to do this, we're going to use two model systems. For prokaryotes, we're going to look at E. coli, and for eukaryotes, we're going to look at the human model system. So let's get started. We'll start with an overview of the regulation of transcription. Now, the expression of genes refers to how a specific gene is transcribed to yield RNA. Those genes that are highly expressed are highly transcribed. In many cases, this RNA is translated to protein. The expression of genes is temporospatially regulated, and what this means is that genes are regulated differently at different times in different spaces. So the needs of the organism changes with developmental state. As an organism grows, their needs change. Organisms must also be able to respond to different environmental cues by upregulating or downregulating genes to more effectively utilize those things that are available in the environment. Additionally, genes are differentially regulated according to tissue type and cell type, and it is these differences in gene regulation that allow cells to differentiate and to function properly. Now, the regulation of transcription happens differently in prokaryotic systems and eukaryotic systems. This is due to differences in genome organization, in RNA polymerase, and in differences in the organism themselves. Eukaryotes have a very complex chromosomal structure in which the DNA is tightly packed, first into nucleosomes and then into solenoids. One of the jobs of the transcriptional machinery is to get into the target DNA, but it has a hard time doing that if the DNA is so highly packed. And so a lot of transcriptional regulation in eukaryotes is just providing accessibility to the transcriptional machinery. Now, with gene organization, that's different in eukaryotes and prokaryotes as well. Prokaryotes try to maximize efficiency. It's one cell that has to respond to a changing environment rapidly and efficiently. And so in many cases, genes that operate within the same system, for instance, the utilization of a specific food source, are organized in a specific stretch of DNA that's turned on all at the same time, so that the genes necessary for that particular pathway are all turned on concurrently. There are also differences in RNA polymerase, in how the polymerase is targeted, and in the structure of the RNA polymerase itself. Now, when we looked at RNA polymerase in prokaryotes, we saw that it is a heterotetramer. It is made up of two alpha subunits, a beta prime, and then a sigma factor. The sigma factor actually targets the RNA polymerase to the site of transcription. So it is the polymerase itself that helps to target the promoter. But with RNA polymerase in eukaryotes, there are several proteins that are involved in targeting the RNA polymerase to the start site of transcription. And so the targeting does not come from the polymerase itself, but rather it comes from a whole host of accessory proteins that help localize the polymerase to the start site of transcription. There are also differences in the organism itself. Prokaryotes must respond efficiently and effectively to changes in the environment, and they must be able to utilize things from their environment efficiently so that food sources and energy sources are not wasted. They also might find themselves in a particularly inhospitable environment and have to regulate gene transcription based on that. Now, with eukaryotes, and especially higher eukaryotes, there are specialized cells that carry out specialized functions, and so it's necessary to have specialized gene transcription in those particular cells. These cells must be able to respond to changes in the environment as well. Another major effect is that prokaryotes do not have a nuclear membrane. Transcription and translation can happen concurrently, and it's more difficult to separate transcription factors from the DNA itself. Whereas in eukaryotes, because there is a nuclear envelope, it's easy to be able to exclude transcription factors from the nucleus, and thus help to regulate transcription in that way. Also, RNAs that are made in the nucleus must be transported out of the nucleus for translation. These figures are from a paper by Gary Stroll. On the left, we see panels that look at the differences between chromatin organization in prokaryotes and eukaryotes. Now, with prokaryotes, the chromatin itself is generally not restrictive. And so, by removing a repressor, the RNA polymerase can have access to the chromatin. And so, how well that particular piece of chromatin is used depends on activators that can help to bind the polymerase to the site of transcription, or it also can depend on the activity of the promoter itself, on how close to the consensus sequence that promoter is. And again, the polymerase is targeted by the sigma factor. Eukaryotes have a significantly more difficult time. The repressed state, or the silent state, can include repression by accessory proteins and also by chromatin modifying activity. The silencing of genes involves repressors that recruit chromatin modifying proteins that, through modification of histone tails, change how histones interact with the DNA and with each other. This can effectively close down the gene, preventing the RNA polymerase from getting to the target. Once the repressor and the chromatin modifying protein are gone, the gene is still in the restrictive state, and this is because the chromatin is tightly wound around histones. What needs to happen is that activators, which recruit chromatin modifiers, modify the histone tails in the nucleosomes surrounding the gene's promoter, and thus allow the polymerase. And at this stage, the gene is at what's called a poised state. The promoter itself is accessible, but the transcriptional factors have not bound to the site of transcription yet. Those proteins that help to target the polymerase itself to the start site of transcription can further activate these genes. So with eukaryotes, the ground state is still restrictive. The DNA is wound around nucleosomes. We can further make this even more restrictive by modifying the histones to make them more compact and more difficult to unwind. This is caused by modifying the histone tails and also by having proteins that bind to the chromosome in this state. In the panel to the right, we see differing stages of chromatin accessibility. In the first stage, we have an activator A that is going to recruit chromatin modifying activity. These are enzymes that modify histone tails. Now, in so doing, these modified regions of the DNA can now act to recruit more chromosome modifiers. And so in this way, we can have a localized remodeling of the chromatin to allow greater accessibility to the gene itself and to the promoter region. Finally, an activator B attracts TF2D and other proteins involved in targeting the polymerase to the start site of transcription. And so at each one of these steps, 1 through 6, there is just an interplay of different proteins and different regions of the DNA. And so by modifying what proteins are available and what regions of the DNA are accessible, you can have a really tight control on which genes are expressed and at what level those genes are expressed. So let's talk a little more specifically about these steps. Now, when we're looking at scenarios like we saw in the right-hand panel, what we're looking at is the interplay between cis factors and trans factors. Cis factors are actual DNA sequences. These may be promoters or enhancers and silencers. A silencer is a region of the DNA to which a repressor will bind to downregulate transcription from that gene. Silencer and enhancer elements are cis regulatory elements that are outside of the promoter region. These may be several base pairs, several hundred base pairs, or thousands of base pairs away from the start site of transcription. These regions of DNA are sites for transcription factor binding. Now, these transcription factors themselves are trans factors. These are proteins that regulate transcription. And they're abbreviated TF. Repressors are downregulators of gene activity. They bind to silencer elements and work to decrease the use of a particular promoter. Activators, on the other hand, bind enhancer elements and activate transcription from a particular promoter. Now, transcription factors must make DNA-protein interactions that are a structural and chemical match. As for the structural match, transcription factors must recognize and bind in the major groove or the minor groove, or both. As far as the chemical match, transcription factors must have residues that can make the right contacts with the DNA in that specific region. So they must have a protein structure that will be able to make contact with the DNA in a specific region, first recognize that DNA, and then bind effectively. Now, many silencer and enhancer elements have an intrinsic dyad axis of symmetry, and what this means is that there will be an inverted repeat on either side of the plane of symmetry. And this is a common feature in enhancers and silencers. Transcription factors have two major domains, a DNA binding domain and an effector domain. The DNA binding domain, abbreviated DBD, can be helix-turn-helix, beta-zip, or zinc fingers. These are the predominant types of DNA binding domains, and we'll look at each of these in just a minute. Now, effector domains can either activate or repress transcription. These effector domains can recruit other proteins, or can interact with the polymerase II itself, or they can interact with what's called a mediator protein. Mediator proteins serve as a bridge between the transcription factor and polymerase II. In this figure, we see a helix-turn-helix DNA binding motif. One helix is going to recognize and bind in the major groove, and the second one is going to pack the first helix in place by making interactions with the phosphate backbone. Helix-turn-helix motifs are another name for the helix-loop helix motif. Different name, same motif. These motifs figure highly in transcriptional regulation in prokaryotes, and in this diagram we see three proteins that all have helix-turn-helix DNA binding domains. These are the LAC repressor, the CAP protein, and the TRP repressor. The LAC repressor and the TRP repressor both downregulate gene activity, whereas the CAP protein upregulates gene activity. The helix-turn-helix motifs found in these proteins are highlighted in yellow. Eukaryotes also have helix-turn-helix motifs as DNA binding domains, and here we see the DNA binding domains of homeodomain proteins. These sit in the major groove in a similar fashion to those that we saw in prokaryotes, with one helix binding in the major groove and another one being held in place by interactions with the phosphate backbone. One thing that I'd like to draw to your attention is that homeodomain proteins can form homodimers or heterodimers. Because we can make homodimers or we can make heterodimers, we can affect the sort of activity that these particular transcription factors will have on the genes that they regulate. By expressing specific types of homeodomains in specific cells, you can change the expression profile of genes in that cell. Homeodomains figure prominently in the establishment of cell fate during early development. They're one of the prominent transcription factors that help to establish body plan in the developing embryo. And again, by being able to interchange differing types of homeodomain partners, you can modulate gene activity differently depending on the types of homeodomain proteins that are expressed in that cell type. In this diagram, we see another type of DNA binding domain that's also found in eukaryotes, and this is known as a basic leucine zipper. Another name for this DNA binding motif is a B-zip. Now, basic leucine zippers, as the name suggests, has a leucine zipper portion and a basic region. These alpha helices are amphipathic, and they can dimerize through the leucine zipper region, which contains a hydrophobic patch that's found on one side of this helix. So each of these helices has a hydrophobic face and hydrophilic face, and the hydrophobic faces serve as a dimerization motif, as those hydrophobic surfaces seek to bury themselves together to avoid the hydrophilic aqueous environment. And again, these amphipathic helices make coiled-coiled interactions in which the hydrophobic leucines bury themselves to avoid the aqueous environment. Now, leucine zippers bind to DNA through the basic region, which inserts into the major groove. By being able to form heterodimers and homodimers, again we get a wide array of binding partners, and we can modulate gene activity by expressing differing types of binding partners within specific cell types, or at certain times in development, or within certain tissues. And again, these proteins form dimers through interaction between the zipper region within the DNA binding domain. I would like to remind you that what we're not seeing is we're not seeing the effector domains of these proteins. We're just seeing the DNA binding domains of these proteins. Another DNA binding domain that figures prominently in eukaryotes is the zinc finger. As the name suggests, zinc fingers bind zinc, and in so doing, form a looped structure that looks like a finger that then inserts into the major groove to hold the DNA binding domain in place. One of the zinc finger motifs is a cis2 his2 zinc finger. The zinc is coordinated by 2 cysteines and 2 histidines to form a finger-like projection that can insert into the major groove. In many cases, the zinc ions are coordinated by 2 cysteine and 2 histidine residues. However, other zinc fingers can be coordinated by 4 cysteines. Now, some transcription factors have several zinc fingers, but others, like the glucocorticoid receptor, have only 2. Some transcription factors, though, have several zinc fingers that actually follow the major groove for a long stretch of the DNA. So now let's take a look at transcriptional regulation in prokaryotes. And to do this, we're going to use the lac operon as a model system. Now, prokaryotic genes are often organized in operons. The genes that are involved in the same pathway will be organized into a specific block of DNA so that they're all utilized at the same time. And this is known as an operon. The lac operon is shown below, and it contains 2 promoter regions, one that affects the synthesis of the repressor, the inhibitor messenger RNA. This gives rise to the lac repressor protein. This lac repressor protein downregulates transcription from the promoter just downstream. This is shown as the orange P region. This regulates the transcription of the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. Now, these 3 gene products are synthesized as one long transcript, the LAC mRNA. This means that the LAC-Z, Y, and A genes are polycistronic. They're synthesized in one long RNA that gives rise to multiple proteins. The LAC messenger RNA gives rise to beta-galactosidase, which is the LAC-Z gene product. Permease, which is the LAC-Y gene product. And transacetylase, which is the LAC-A product. These 3 gene products, beta-galactosidase, permease, and transacetylase, are used in the utilization of lactose as a fuel source. Now, these 3 genes, the LAC-Z, Y, and A genes, are regulated by the promoter and the operator region that are just upstream of these genes. Now, the LAC-I gene encodes a message that gives rise to the lac repressor, and the lac repressor binds to the lac operator. As it does so, it prevents utilization of the lac promoter. This prevents transcription of Y, C, and A genes. Now, the repressor can be bound by an inducer. Now, this inducer is going to induce transcription of the lac messenger RNA, thereby allowing production of beta-galactosidase, permease, and transacetylase. This inducer binds to the lac repressor and reduces the affinity of the lac repressor for the operator region. The repressor-inducer complex does not bind DNA effectively, and so this allows the promoter region to be used to synthesize lac messenger RNA. Now let's go over again what lactose is. Lactose is a disaccharide that's linked together by a beta-galactosidic bond. This is a beta-1,4 linkage between galactose and glucose. Now, the beta-galactosidase gene is going to hydrolyze this beta-1,4 linkage to liberate galactose and glucose. Now, 1,6-allolactose is a metabolite that's produced from galactose and glucose. It's a beta-1,6 linkage between the 2 monosaccharides to yield the disaccharide. This is a potent inducer of the lac operon. It will bind to the lac repressor, change its conformational shape, and prevent it from binding to the LAC operator. This leaves the LAC promoter available for transcriptional initiation. Now the thing is once you leave the promoter open, certainly this gene is now available for transcription, but it's not activated. It's not going full force. You can increase the rate of transcription from this promoter by increasing the affinity of RNA polymerase for this promoter. Now the thing is, the LAC promoter is not the best promoter, but it can be made more effective by a transcription factor. In this case, the transcription factor is the cap protein. This is the same protein that we saw before that has a helix turn helix DNA binding motif. The cap protein is activated through the binding of cyclic AMP. And the name cap can refer either to catabolite activated protein or cyclic AMP activated protein. I've seen both used. What's going to happen is, when glucose is not readily available, this stimulates the production of cyclic AMP. Cyclic AMP is going to bind to the cap protein, and in so doing, it's going to increase its affinity for DNA. It will bind to the DNA at the LAC operator and recruit RNA polymerase to the start site of transcription. In this way, a decrease in the concentration of glucose available for utilization is going to activate an open LAC promoter. Now I say open LAC promoter because you need two things. You need for glucose levels to be low, and you need for there to be lactose in the environment for the LAC operon to be maximally activated. So let's look at this a little more closely. Here we see a repressed LAC operator. The LAC repressor is sitting at the operator region. In this state, the LAC operon is repressed, and beta-galactosidase, permease, and transacetylase are not going to be synthesized. When there's lactose in the environment, allolactose is going to be made, and it's going to bind to the LAC repressor. It is going to reduce its affinity for the operator sequence, and it's going to fall off. The operon is no longer repressed, but transcription is low at a basal rate. The operon is still not activated. Decreasing glucose concentrations results in an increase in cyclic AMP. This cyclic AMP is going to bind to the cap protein and increase its affinity for the promoter region. Because the cap protein is in place, it serves to recruit the polymerase to the start site of transcription. It stabilizes the interaction between the sigma factor and the promoter so that this promoter can be used more effectively, and so transcription of the LAC operon is activated. So now let's change gears and look at transcriptional regulation in eukaryotes. We're going to do this by looking at the estrogen receptor. Now, estrogen receptors bind estradiol, which is an estrogen. Because this is a sterol, it can diffuse through membranes, and the estrogen receptor is found largely in the cytoplasm. Upon estrogen binding, the estrogen receptor is translocated to the nucleus where it binds to specific regions of the DNA known as estrogen response elements. From this position, the activated estrogen receptor can have an effect on DNA and transcription. Now, other estrogen receptors are already at the nucleus, sitting at the estrogen response elements, even though there is no estrogen bound. These estrogen receptors are poised and ready for estrogen binding. and then they will have an effect on DNA and on transcription. Now, the estrogen receptor has a DNA binding domain and a ligand binding domain. The DNA binding domain is a zinc finger that inserts into the major groove. The ligand binding domain has a ligand binding pocket near which is a specific helix. This helix changes its position upon estrogen binding. Estrogen receptor binds DNA as a dimer, and the dimer is shown below. You can see that without estrogen in the binding pocket, helix number 12 is extended outward, and when estrogen binds, this helix changes its shape and the estrogen receptor becomes more compact. The estrogen receptor exerts its effect by attracting coactivators and corepressors. Some tissues express coactivators more highly than the corepressors, and so when estrogen binds to the receptor and causes a conformational change in the shape of the ligand binding domain, a coactivator can bind to this domain and then can remodel chromatin to make it more accessible. And this increases transcription from that gene. However, some cells express corepressors more highly than they do coactivators. And upon estrogen binding, the conformational change occurs, but this time what's attracted to the ligand binding domain is a corepressor. This corepressor is going to have a negative effect on genes, decreasing their rate of transcription. Tamoxifen is an estrogen analog. It binds to the estrogen receptor, and when it does, it prevents helix 12 from folding down correctly, and so the estrogen receptor does not undergo its normal conformational change upon tamoxifen binding. Tamoxifen prevents estrogen binding. The ligand binding domain does not change shape, and so this estrogen receptor is not going to attract coactivators or corepressors. Now, tamoxifen, therefore, is going to act like an antagonist in some tissues. These tissues contain coactivators. Tamoxifen will bind in the ligand binding domain and prevent estrogen binding. The estrogen receptor is sitting on the DNA in the off position, and that gene will be transcribed at a basal level. So, tamoxifen prevents activation, and these genes cannot be activated by the coactivators. On the other hand, tamoxifen can act like an agonist in other tissues. These are the cells that contain the corepressors. Tamoxifen is going to prevent the repression, and these genes will be transcribed at a basal level. This occurs in bone tissue, and tamoxifen is often used for osteoporosis therapy. Because tamoxifen will bind to the estrogen receptor and prevent the shape change and prevent the corepressors from binding. And so in closing, in eukaryotes, transcriptional regulation depends on what sort of transcription factors are present in the cell and what sort of signals the cells get. The same signals can have markedly different effects on different cell types because of the transcriptional regulators that are present. This marks the end of Lecture 38. Have a great day.